This is lesson 2 for the upper sixth, architecture and control of gene expression as an introduction. This follows on directly from lesson 1, which was the PowerPoint on or introducing the idea of genetic control circuits and the different levels at which we can regulate gene expression, both at the transcriptional level and at the translational level. So these are the objectives for this particular lesson. We're going to look at the component parts of a gene, sort of a generic gene, I might add. We'll get to differences between prokaryotic and eukaryotic genes in a minute. We will look at then the overall sort of gross architecture of a generic eukaryotic protein coding gene. We will then look at, do a quick recap, and I aim for it to be reasonably concise, as a sort of overview of transcription, which you covered last year in the lower sixth year. We will then look at the sort of upper sixth bit of that, which is looking at how the nascent transcript, the pre-mRNA, is modified by your cells, by eukaryotic cells, specifically, to then become mature messenger RNA that is ready for translation. Finally, we will then look at the bacterial gene and its architecture on the chromosome. So let's kick off with objective number 1, which I'm going to change to purple because it's regal. And we will look at the components of a gene. So component 1 is just the generic components of a protein coding gene. And I make the point about protein coding because, of course, at A level, we now know the definition of a gene. Just to remind you, always good to link back to things. I know it makes the videos a little longer, but I feel as it's useful to link back to these things, is a sequence of bases, specifically in DNA. Genes do not exist in RNA per se. It is referring, a gene refers to the sequence of bases in DNA that code for, this is the sort of A level bit, or a functional RNA molecule. That is our definition of a gene at A level. Now, the honest truth here is you still could get away with just going up to here and just saying that it's a sequence of bases that codes for a protein. And that would serve you fine. I make the point about the functional RNA molecule because, of course, the molecules that you came across last year that are RNA coded by the genome, they have to themselves have been transcribed. But they don't get translated. They remain as functional RNAs. The examples you came across, of course, were the tRNA molecules, the tRNA adapters in translation, and the ribosomal RNAs are RNAs that make up the ribosome. There is another set of RNAs called small. Well, we'll get to it. You don't need to know them really, but I'll mention them later. There is another third type called snRNAs, and we'll talk about those later. But these are the key 2 ones you need to know for your A level. And of course, they are not translated into protein. They don't make a protein. They carry out their function as they are. But of course, they're transcribed from a gene. Right. So let's have a look at these components. So all genes must have the following. So if I draw my DNA like this, I will just draw one strand for now. Imagine this is the double helix. This is two strands. I'm just going to draw it as a line. And of course, you have your polarity, so 5' and 3'. You have a gene. So let's sort of put the gene in here, and I'm going to code it in blue. So this here in blue, this sort of box, is a gene, protein coding gene, let's just say. And let's say you've got another one in purple, slightly smaller gene here. So let's just say there are two genes on this particular bit of DNA that we're looking at. And of course, they're both going to code for proteins. This one might make an enzyme here, and the blue one might make an enzyme that looks like this, for example. Two separate enzymes, two separate genes. The key thing is to understand that although I've color-coded them in boxes, in real life, this is just a sequence of A's, T's, G's, and C's. And the trick for transcription, because don't forget, we want to make the intermediate step here is to make an mRNA molecule that corresponds to this gene here, and one that corresponds to this gene here. We do that by the process of transcription, and we need to recruit RNA polymerase at the start of each gene to begin transcription. And we have to tell it which way to go. And that is achieved of the start of the gene. I will draw it as a circle here. So we'll have one here in front of the blue gene, 5' end, and we'll have one in front of the purple gene here. And these are called promoters. The function of the promoter, if I write that here, is essentially it is the binding site for the enzyme that's going to carry out transcription, which is RNA polymerase. Easy to remember because it's a polymerase, so it's going to take loads of monomers and put them together to form a polymer, and it's going to do that with RNA nucleotides, ribonucleic acids. Okay, so essentially, transcription will start here, and promoters, interestingly, contain directionality. So within this sequence, it actually contains directional information. Directional info, which I've alluded to there with that blue arrow. What that means is when your RNA polymerase, which I will, what color? Let's go yellow. When the RNA polymerase sits on top, so RNA polymerase is recruited, RNA pol, it knows which way to go. It does not start moving this way along the DNA and making RNA here. It knows, because of the sequence of the promoter, that the gene starts downstream towards the 3' end of the promoter. So therefore, it will open up the double helix using its intrinsic helicase activity. Look back at the lower 6 notes if you need a recap on that. And it will start transcribing, as I've indicated with that black arrow, in the downstream direction, and it will produce this RNA. So if I get the right color of blue, it will start transcribing here, and it will make a copy of the RNA. So it will transcribe a little bit of RNA from this bit of DNA here, and then it will eventually get into the actual coding information of the gene here. The RNA polymerase knows when to stop because, as well as having a promoter sequence that essentially says, you know, bind here and start transcribing in this direction, this is where the gene is, there's a sequence at the end that does the opposite thing. So I'll do it in red because it's a stop. I'll draw it as a triangle. This here is called a terminator. And of course, there'll be one at the end of gene 2 as well. And again, this contains information that when the RNA polymerase reaches here, after it's transcribed our mRNA, the terminator sequence, through mechanisms you don't need to know, tells the RNA polymerase to fall off the DNA duplex and dissociate from it, releasing it into the nucleus. Those consensus elements are repeated for each individual gene. So that is the sort of overview of the protein coding genes. We have promoters and terminators, which essentially act as start and stop signals, and they sort of effectively, in inverted commas, tell the RNA polymerase where to start transcribing and in which direction, and where to stop. Now, just not wishing to confuse you, but actually, genes don't have to face in the same direction. So this purple gene here could actually have the promoter and the terminator at different positions. You could have the promoter here and the terminator here. And of course, the RNA polymerase would be recruited to the promoter here, and the promoter would contain the information saying, go transcribe me this way. So the product would still be made, the pre-mRNA would still be made, and when the RNA polymerase reaches the end here, oops, wrong color, reaches the terminator, it then falls off again. So in this case, we have sort of transcription occurring in two different directions. This way for the blue gene, and this way, hypothetically, of course, for the purple gene. They don't have to be going in the same direction. And it is the promoter, because it is directional, it contains directional information, that determines which way the polymerase will go. But that is not required for A level. But it's an interesting point, nevertheless, I feel. So let's now look at, so that's number one done. Let's now look at the eukaryotic gene architecture. So I'm going to make this a little more complicated now by looking specifically at a eukaryotic gene, protein coding gene. We don't talk about the non-protein coding genes. So, where's the black pen? Here we go. So this is our gene again. We're going to build our gene. So I'm going to kick off with a promoter in green. Here. So I'm going to label that. And I'm going to label the terminator. So the gene, unlike how I've shown it here, where it's one continuous block of coding information in blue, and actually I should just point out here that there'll be a little bit of transcription for this bit in here, of course, before it falls off. So the RNA actually has a little bit of additional sequence either side of the protein coding bit. Unlike how I've shown up there, where the gene is a continuous string of coding sequence, in eukaryotes, this sequence is actually interrupted. Bits of DNA that do not code for the protein, shown in blue here, actually insert themselves within the coding sequence. And what that looks like, if I take the blue gene here as an example, what that means is we have extra stuff, let's say I do it in orange here, extra bits of DNA that get sort of inserted into regions of the coding sequence here and split up the coding sequence. So it would look like this. So if I go back to blue for our gene, let's say this is the first bit of the gene, the first few hundred base pairs of the gene, but that's been interrupted by, we'll just keep it in black here, by a bit of DNA that doesn't actually code. It doesn't contain any useful information. Well, that's a little bit of a simplification, but for our purposes, for coding for proteins, it does not contain the codons required for the correct amino acids for our protein. The remainder of the gene might be split up here. You might only have maybe 50 base pairs or something before it's then split up again into another section. And these vary in size. And then you might have the rest of it here before you get to the terminator. So it's not that the gene has disappeared or is being taken away. It's being literally interrupted by additional bits of DNA that insert themselves between the bits of coding information that are actually useful. So if this is DNA, just to be clear, our start codon will be right at the start of this first block, which of course is going from 5' to 3', A, T, G. And our stop codon, one of the three, will be at the end of the last bit of coding sequence, which again, 5' to 3'. I'll do it bigger actually. So 5' to 3' will be no, it won't. It will be T, A, G, T, G, A, or T, A, A, going from a 5' to 3' direction. That's not a very good diagram, but hopefully that makes sense. And these, of course, are the codons. So if I do that in purple here, this is the start codon, which is different to the promoter. So the start codon, don't forget, is the sequence, it's the triplet of bases in DNA or the codon in RNA that is the first amino acid that the ribosome is going to put in. So ATG codes for methionine, which will be the first amino acid placed into the protein primary sequence. The stop codons come in 3 flavors here, and they essentially tell the ribosome to dissociate. They have no amino acid that they code for, so the ribosome dissociates. So that's for translation. So the codons, bear in mind these are the stop codons, are for translation, not to be confused with the promoter and the terminator, which act during transcription for RNA polymerase. So just to be very clear, the RNA polymerase, I think I used orange last time, RNA polymerase binds at the promoter, so this is RNA polymerase, and the ribosome, once we've dealt with the RNA, will bind, if I do that in, we'll do it in like the starry ocean color, whatever that's called, the ribosome will bind to the start codon there, and will sort of dissociate out the stop codon. The only other thing to mention here is that the coding sequence here in blue, these bits are referred to as exons. And I remember this because exons are expressed as in they code for protein. That's the actual useful codons that contain the coding information to make our protein product and put the right amino acids in the correct order. The intervening bits of DNA, which just for the sake of ease, I will highlight in purple here. So we've got one there, and one there. These intervening sections are called introns. And they are called introns because they interrupt, although that's how I remember it. They interrupt the coding sequence. They're not useful to us. So this is the overall structure of a eukaryotic protein coding gene. a little more complicated than just what we assume at sort of GCSE, which is that you just have one continuous piece of DNA that codes for a protein. It is not continuous. So I think that's all I wanted to say with regards to the eukaryotic architecture. So we've done point one, eukaryotic architecture we've done for point number 2. So we're going to cover now briefly an overview of transcription. So here we go, point number 3, which is transcription. So very quickly, what is going to happen, and I can probably use this diagram here. If I get rid of some of the stuff beneath, is as follows. So if I get rid of that ribosome there. What happens, first of all, from last year, is that RNA polymerase binds to the promoter region. How it does that, we will come across in a few lessons' time. So RNA polymerase, in orange here, binds to the promoter. Don't forget the promoter contains the directional information to start transcribing from left to right, in this case, from 5' to 3'. Once the RNA polymerase is bound to the promoter, the next step is that the RNA polymerase, I'll just abbreviate it to RNA pol, uses its intrinsic helicase activity to break hydrogen bonds between base pairs, between the 2 strands of DNA, and this generates what we refer to as a transcription bubble. So in the local environment of the 2 double-stranded bits of DNA, we form this. The base pairs, which I will do in green, don't forget hydrogen bonds between the bases, between the 2 different polynucleotide strands, are broken. And the bases in question, of course, are sticking out here, like that. And of course, RNA polymerase is sat directly on top. Feel free, nota bene, to make a sort of additional note here. What I mean by this intrinsic helicase activity is that a separate DNA helicase is not needed. So no sort of separate helicase is required. RNA polymerase does it itself. Now, there's a sort of far more complicated answer to this, which I won't bore you with now. I might mention it when we look at eukaryotic transcription, for example. But for now, feel free to remember it as RNA polymerase looks like this, and it has a little part of it here that is capable of helicase activity. Remember, helicase is not one enzyme, it is a family of enzymes that can use ATP hydrolysis and the energy from that. The energy liberated from this, so energy here, is used to translocate along a strand of DNA and break hydrogen bonds between the base pairs. Look back at DNA replication if you want more information on helicases. But that's essentially what it does. So it opens up in a local region here. That's important. This whole region here is local. It doesn't do it for the whole strand. It just does it initially here. What we then have is floating around in the nucleus, we have our nucleotides. But don't forget we're making RNA, so they're ribonucleotides. They're not deoxy. They have an oxygen on both the 2' and the 3' carbon. So we call them activated, because they have 3 phosphate groups, RNA nucleotides. Activated RNA nucleotides diffuse randomly into the RNA polymerase entry channel. I'll explain this in a minute. And hydrogen bond with their cognate, their partner, their complementary base pairing, their cognate base on the template strand. Of DNA. Okay, remember, only one of these strands is going to be copied. So when we make the RNA, look, the nucleotides are going to come in. They actually come through the entry channel in the polymerase here. So the nucleotides diffuse in down here. If they're correct, they will form hydrogen bonds here between their, using complementary base pairing. And if correct, then phosphodiester bonds will be formed between them, and you make your sort of nascent RNA molecule. But the key thing to remember is we copy only one strand. Unlike replication and DNA polymerase, RNA polymerase in orange here will only copy one strand. And it copies, in this case, this strand, which we call the template strand. The strand that isn't copied, in this case the top one, is called the coding strand. And if I'm not going to, sort of for the sake of time, cover that. It will be in a different video in a lower sixth topic when you do transcription in lower sixth. You should know the difference between the template and the coding strand. Just as a little bit of information here that you should already know, the coding strand is always the 5' to 3' strand as you look at it, and the template strand is always the 3' to 5'. So that's what I mean here. So the activated RNA nucleotides, by the way, I think it's important just to recap this bit. They are activated. You have to call them this, because what we mean by activated is that you have your 3 phosphate groups attached to your ribose sugar, which is then connected to one of the bases, right? Either A, U, G, or C. Uracil swaps for thymine, of course, in RNA. And it is not deoxyribonucleic acid, it is just ribonucleic acid. So therefore, on the 2' carbon here, we have a hydroxyl group, and on the 3' carbon, we also have our hydroxyl group. If it was a DNA nucleotide, then of course this hydrogen, this oxygen here would not be there, and it would just be a hydrogen. There are 3 phosphate groups here, so these are inorganic phosphate groups. And essentially it is these bonds, these two here, which are phosphoanhydride. Bonds that contain the high energy bonds that when we hydrolyze them, when RNA polymerase hydrolyzes them, that provides the free energy. Oops, I don't want a calculator. The free energy required for catalysis. And again, if you want to look at the structure of those bonds in more detail, look back at the ATP and transcription lessons from lower sixth. Hopefully that shouldn't be too new to you and should make sense. So our activated ribonucleotides, RNA nucleotides, not deoxy, diffuse into the RNA polymerase entry channel. They hydrogen bond with their cognate base via complementary base pairing. Don't forget, if this is an exam answer, be specific, via complementary base pairing. Always state it when you say this as well, just in case. So I would always say that the A will obviously pair with the U, and the C's pairs with the G. 2 hydrogen bonds for A's and U's, 3 hydrogen bonds for G's and C's. Always worth mentioning that when you talk about complementary base pairing. Very often in mark schemes, you get marks for citing the rule. Anyway, so once this happens, if the base pairing is correct, then RNA polymerase will catalyze the formation of a phosphodiester bond. And again, that should be, you should know this, that it is using phosphates and there's a diester bond. In other words, there's sort of two ester linkages there that connect the phosphate group either to another phosphate group or between a phosphate group and a sugar. And it's worth pointing out, of course, that this is ribose sugar, just in case we have forgotten. This is ribose, not deoxyribose. And essentially, this continues. And I'm just going to make the point, actually, here, that the free energy required to do this reaction comes from hydrolysis of your 2 phospho anhydride bonds between the pi groups, between the inorganic phosphates here. And obviously there's a third one, but don't worry too much. What's released is something called pyrophosphate. You actually release 2 inorganic phosphate molecules because it is a condensation reaction. And again, I realize I'm going into a lot of detail, but I hope it's a good revision resource for you when you're watching this video. Okay, so again, look back at the lower sixth if you're not sure. Very quickly, we'll just do the mechanism because I think it is important. So let's imagine we have our activated ribonucleotide. So we've got inorganic phosphates here. These are our 2 high energy phosphoanhydride bonds. This then connects, of course, to our ribose sugar here, 5 carbon sugar. Don't forget, carbon 1, 2, 3, 4, and 5. And this then connects to the bases, the organic nitrogenous bases. Do not just say base. So I'm going to change that. It's an organic nitrogenous, I will just shorten as N, base. Okay, of course, the important groups we know are that on the 3' we have a hydroxyl group, and because it's RNA, we do actually have one on the 2' as well. We're not going to worry about the 2' one for now. Don't forget, this is the 3'. So this is 1', 2', 3', 4', 5' carbon. And let's imagine we've got a nucleotide that is already in DNA. So let's just, I'm just trying to figure out how to draw this now, actually. Maybe I should have drawn the phosphates. No, okay, we'll keep going. Importantly, you have your 2 lone pairs on your oxygen here. And obviously, linking back to lower sixth, the oxygen is delta negative. If I do that in red, and the hydrogen there is delta positive. The reaction that happens is that if this is the free ribonucleotide and we look at a nucleotide that's already been incorporated into RNA, so let's draw one. I'll keep in black, but this is in the RNA molecule itself. We have here, we want to attach this to the RNA molecule. My laptop's done something a bit weird. Not sure what's going on here. 2 seconds. Oh, we're back, I think. I hope that's still recording. If not, this has been a colossal waste of time. Right, I'm going to keep going anyway. I'm talking to myself now. Well, I'm talking to myself anyway, but anyway, enough rambling. Right, I hope this is still recording. I think it is. I've just checked. So this is the ribonucleotide triphosphate that is incoming into the RNA strand. And of course, we have a nucleotide that's already in the strand. So let's just draw that now. I'm going to do it a little bit smaller, but same principle. We've got our hydroxyl groups. Our phosphate there, which obviously is now is sort of linking to elsewhere in the chain. And we're going to add our nucleotide in, this nucleotide triphosphate here. So this is the rest of the chain of RNA that we've already made. So you can imagine it just continuing. It's important to label our carbons. So this is 1', 2', 3', 4', 5'. And of course, the golden rule is that DNA replication will proceed in a 5' to 3' direction. The reason why you don't need to know is it is interesting, so do ask me, but I'm not going to cover it in this video because you don't need to know it. But the polymerase essentially is sat at the end of the growing chain. So the polymerase is sat here, not to scale. And of course, the entry channel is here, and this nucleotide here is going to diffuse into the entry channel like that, and the reaction is going to happen, the condensation reaction. So I'm going to get rid of the polymerase for now, but it is there. The condensation reaction happens like this. Let's put our lone pairs in. It's the 3', don't forget, 5' to 3' direction. So we're going to have to add at the 3' end. And of course, don't forget, this is the 5' carbon here. Whoops. My surface is being a bit weird, but anyway. So this is the 3' carbon. This is the 5' carbon. The connection is essentially going to be made in a 5' to 3' direction. So how does this work? The lone pairs, don't forget this harks back to lower sixth, the lone pairs of electrons on that oxygen there can act as nucleophiles. It's a nucleophile. And of course, the phosphate group here, if I were to just sort of zoom in on what this looks like, it looks like this. And then this connects to the next phosphate group. And hopefully what you appreciate is all of these oxygens are delta negative. So it's going to be pulling, all of these are going to be pulling electron density away from the phosphate group. So that means the phosphate in the middle is delta positive. So if I zoom in here, this phosphate is delta positive. It is an electrophile. So what you can hopefully appreciate happens is that the lone pair of electrons, don't forget I'm going to draw electron diagrams here. So a double arrow refers to the movement of a pair of electrons, and a single-headed arrow refers to the movement of just a single electron. So we're going to do double-headed arrow. Both of these electrons here are going to attack the phosphate group, specifically the phosphorus atom, which is an electrophile in the middle here. And it's going to form a bond with that phosphorus atom. What then happens is this particular bond, the 2 electrons in this covalent bond, then break off essentially and get given back to that phosphate group. So both of those double-headed arrow move, which means that you have the net result of forming this bond. So if I get rid of the electron movement sort of diagram here, these 2 blue electrons, which are shown blue, actually form a sort of covalent bond. Technically, it's a dative covalent at the moment. These 2 electrons form a dative bond or covalent bond to that phosphate group. This bond here is broken, so I'm going to get rid of it. And then eventually this hydrogen gets taken away by water, but you don't need to worry about that. So what you end up is with this. Those electrons are now here in this covalent bond, and we have released our molecule of 2 phosphate groups, and it is called pyrophosphate. Now this is a condensation reaction. And don't forget, this is it's one of the examples in biology where in a condensation reaction, it's not actually water that's produced or liberated from the reaction. In this case, it's pyrophosphate, but it is still a condensation reaction. You're still taking 2 smaller molecules, joining them together to form a larger molecule, and you're releasing a small molecule as a leaving group. So that's essentially what happens. Where do we get to? So that's what happens. So phosphodiester bond formation via the nucleophilic attack. And then finally, RNA polymerase reaches the terminator. And through a variety of mechanisms, maybe you'd remember from the lower 6th material, in bacteria, one of the key ways that this is done is via the Rho helicase. It's just a name. It's just a name for a helicase that essentially helps in termination. There are other mechanisms as well, and in eukaryotes it's slightly different, but don't worry about it. Essentially, RNA polymerase reaches the terminator and dissociates, falls off. And that releases the RNA. So that hopefully is just a sort of, it was going to be a brief overview, but as with everything, it's now turned into quite a long overview, but I hope it's useful, linking back to some lower 6th biology, for an overview of transcription. We're now going to talk about the post-transcriptional modification, which is bullet point number 4. So if I can get this, I'm going to see if I can copy this so I don't have to draw it again. Copy. By some miracle, this looks like it's actually working. Of course, it doesn't work now. Right, okay, bear with. Draw it again. Feel free to skip this if you don't want to watch me drawing. But so we've got a promoter, green. We have our terminator in red. And we have our gene in the middle. Let's go for purple this time. That is split into exons that have coding information. They are expressed, and introns. So let's say there's 3 exons. And the introns I'll highlight in purple again. So the ATG, don't forget, is here, start codon. These are the stop codons over here, 1 of 3. So RNA polymerase binds, let's do that in pink, to the promoter, and it will initiate transcription from the promoter. But there's a gap before we get to the ATG start codon. So we're going to have a little bit of transcription, which I will do in black here to reflect that sort of bit of sequence in the middle. So we will have a little bit of transcription of DNA there that contains other sequence before we get into our sort of protein coding bit where we have a bit of exonic sequence here, and we then move back into sort of intronic sequence before then getting into our exon here and our intron again and the last exon. But of course we stop at the terminator, so we keep going, keep transcribing until we reach the terminator here, and then we stop and dissociate. So this is our mRNA that's produced, and it is not yet mature because we've got introns in there, so we call it pre-mRNA. mRNA because it's protein coding, messenger RNA. So this is pre-mRNA. This is the 5' end, This is the 3' end, but we still retain our sequences for the start and stop codons. So the ATG here right at the start will be maintained, but of course it's now RNA. So instead of ATG, it is now AUG. And similarly with the stop codons. So let's just pick one. Let's go UAA. Don't forget TAA was the sequence up here, if that's the case. So that's what our pre-mRNA looks like. The other thing that now happens, so the first step of post-transcriptional modification is we have a 5' cap and a poly A tail added. You don't need to know the details here, but I'm just going to draw that here. Essentially, what happens is we have a sequence added. I will choose a different color. Let's go orange. Here at the 5' end, called the 5' cap. It's a modified guanine nucleotide if you're interested, but don't worry about it. It's sort of like a protecting group. And then at the end, we have a poly A tail, which I will just do in yellow here. So if I highlight poly A tail here, and the poly A tail is what it says. There is a string of adenine nucleotides that are added, and I'll just put N here, to the 3' end. That is post-transcriptional modification. And the whole purpose of this is A, for stability. mRNA likes to be degraded. There are lots of enzymes when we eventually, of course, get into the cytoplasm, sort of endonucleases and exonucleases and things. But there's a nuclease enzyme that looks like Pac-Man here. This is a nuclease, which could easily bind to the end of RNA in the cytoplasm and start to chew it up. Similarly, you could have another one that binds to the 5' end here. And the 5' cap and the poly A tail aid stability because they stop this happening. They stop nuclease action. Nuclease, don't forget, would take DNA or RNA like this, bind to one end, and start chewing it up into bits, into specific individual monomeric nucleotides. And we don't want that to happen, of course, because we want to translate our sequence. So stability is the first thing. The second thing is it actually aids in the process of translation. It aids in ribosome binding. And the third thing is actually getting it out of the nucleus in the first place. So it aids nuclear export. Getting the RNA out of the nucleus. The second post-transcriptional modification has to be that we still have the introns here, right? Let's say we've still got an intron here and here, and we need to get rid of them. So the second thing is that introns are removed. This is achieved through an enzyme complex called the spliceosome. And you must know that name. Very important. Oops. Spliceosome. So a huge enzyme complex. So it's an example of quaternary structure in the same way that the ribosome is and the RNA polymerase. Most enzymes exist as quaternary structure, don't forget. They're not just individual tertiary elements on their own. And it is made up of snRNAs, which I briefly mentioned earlier, small nuclear RNAs, and some proteins. But again, kind of like the ribosome from last year, it is these guys, the snRNAs, that can do nucleophilic attack and achieve catalysis. It's RNA that's doing catalysis. Weird, not protein, interestingly. So this is why the spliceosome, as well as the ribosome, are called ribozymes. in case you're interested, because it is the RNAs, in this case, the snRNAs within the spliceosome, or if it's the ribosome, it's the rRNAs, that can do nucleophilic attack because of their chemical structure and therefore achieve catalysis. You don't need to know how the spliceosome works, but all you do need to know, if I'm going to try and do this in the different colors, is essentially it's going to do this. It's going to cut here and here at the beginning and end of every intron, here and here, and it's going to cut out the introns. Here we go. So the introns are now floating here, and it's going to glue these two bits together. It's going to stick them together into continuous sequence. So I don't want to fill them in because that makes it look like I'm adding sequence, but essentially it will form now a continuous RNA. So what our RNA now looks like is this. We have our coding sequence here that's been stuck together. Essentially, this has been stuck together here, this has been stuck to here. We still have our black bits at either side of sort of just upstream sequence. And we have our cap on our 5' end, and we still have our polyase on the 3' end. So this is now our gene, which looks a bit more familiar to us, hopefully. We've edited it, we have got rid of the introns using the spliceosome, and the whole process is called splicing. Hopefully it makes sense. So splicing is the removal, the enzymatic removal of introns, which interrupt their non-coding sequence. And it's done by the spliceosome, which is mainly made up of RNA, weirdly, and a few proteins. It's a big quaternary structure complex, but it's the it's the snRNAs, the RNA component that achieves catalysis here. This is now our final product. This is now mature messenger RNA. We're absolutely happy with this. We can deal with it. Don't forget, we have our start and stop codons. The start codon will be here, AUG. It's RNA now, don't forget. Our stop codon, which I think I said was UAA. So when this whole thing is transported out of the nucleus, right, into the cytoplasm, because don't forget, if we're in a eukaryotic cell, we've got our nucleus here. It's a double membrane organelle. And our RNA, let's just do it in purple, is now here. It has to be transported out into the cytoplasm, where it can be bound by ribosomes and translated into protein. And of course, translation we will look at, we look at separately in lower sixth. And of course, that transport is facilitated by the fact that there's a 5' cap here and our poly-A tail here, as well as maintaining stability here, because our nucleases are in the cytoplasm that really want to attack this RNA. So if your cap is on there and your tail is on there, they are protected from degradation, at least initially, and you can make your protein. Gosh, right, so that's post-transcriptional modification. And I just want to spend 5 minutes or so now looking at how bacterial genes differ. A lot less detail required here. They're a lot more simple. So this is what, bullet point number 5. So last point for today, which is bacteria. Bacterial gene architecture. So same idea, let's build a gene. And I'm going to go reasonably quickly because it's fairly straightforward. Principles are the same. We have a promoter. And downstream of the promoter, we have a gene. Let's say it's a purple gene here. This is gene 1. But immediately downstream of that, we often have other genes. So let's say there's a pink one. And for the sake of, making it a bit more complicated, let's say there's a light blue one here, slightly smaller light blue gene here. And the key thing is that the terminator is, as we know, at the very end. So this is the terminator. So transcription will start at the promoter and it will end at the terminator. And hopefully what you can appreciate is that these three genes are under the control of the same promoter. We have multiple genes under the control of the same promoter. So we transcribe all three. We only need one binding event by RNA polymerase. So let's draw that in orange. One binding event here, and to set it off, transcribing, and we get three gene products, essentially. We get three genes transcribed into messenger RNA. And so the RNA, if I draw it underneath, would look like this. We have a bit of black bit here. Then we go to our purple, so our first gene. Then we have a little bit of intergenic stuff there. Then we go to our pink gene. Then we have a little bit of, again, intergenic stuff, DNA, and then we go for our blue one, and then a little bit of DNA at the end towards the terminator. Key thing, no introns. Hashtag no introns. There are none, right? This coding sequence in purple is perfectly fine for translation. So is the pink one as well. Just going to plug my laptop in. I think it's about to die. I really hope not. There we go. Technical glitch there, but hopefully we're back. So what I was saying is no introns, we're ready for translation. And essentially, if I draw, don't forget, in the DNA up here, we'd have our start. Oops, this isn't going well, is it? That'll do. We have our start codon here, which is I'll do it in purple if I can. ATG. And here will be a stop codon, which I'll just write as stop. Again, for the red gene or the pink gene, we'd have an ATG here, and we have a stop codon at the end, and same for the blue. So they could be translated independently of each other. So if I translate this into RNA, we have AUG here. We've got stop there. AUG here. And then stop. And then in blue, AUG and stop. And I'm just going to delete this for now. So what happens is, this is already mRNA. It's all ready to go. There's no nuclear envelope to get it out of, to transport it out of. There's no 5' cap, there's no poly A tail, and there's no introns anyway. So we don't need to get rid of them. So immediately, even as you're transcribing this, ribosomes can bind. If I do them in orange, ribosome will bind to the end. It will essentially start translation at AUG here. You'll have another one starting here and another one starting here, all at the same time, potentially. And they're going to move in this direction, left to right, from the start codon to the stop codon, which is where they dissociate and fall off. And we therefore make, protein A in purple, we make our protein B in pink, and our protein C in blue. The key point of this is that we have multiple genes under the control of a single promoter. and this is called an operon. Lots of words, it's literally just all it means. We just call it an operon, and this is how bacteria organize their genes, in operons. We don't have individual promoters and terminators for each gene. We place multiple genes between a single promoter and a single terminator, so they can be transcribed all at once, and we can make 3 separate protein products from them very quickly. So hopefully you can appreciate this is all for increased efficiency, because bacteria can't afford, with their short generation time of 20 minutes, to not be efficient. And the reason it's efficient is because of why we do this whole thing. These 3 enzymes here most likely participate in a single sort of the same biochemical reaction, right? Your reaction might be, and we are nearly done, to convert, substance A into substance D over here. But you might have multiple steps. It might have to be converted into B, then into C, and then through a longer pathway into D. And you may therefore require these 3 enzymes for these steps. Converting A to B, you might require the purple enzyme, not necessarily in this order either. So actually your B to C might require the blue one, and then C to D, the final step, might require your pink enzyme. And the overall reaction is that you convert a substrate, let's say it's A, into a product, which is D here. So this is A, this is D, and it could be that D is your end product. This is what you want to produce. But you need 3 separate enzymes here in order to do it. So therefore, it makes perfect sense to use an operon and to put all those 3 genes under the control of the same promoter. So just scrolling up to the top, I think we have now covered bacterial gene architecture, essentially operons, and how they work.